This lecture talks about the comparison between DNA and RNA. Remember that we've talked about the nucleus of a cell, which is represented by this purple structure here, and those holes represent the nuclear pores. Inside the nucleus, we find both DNA and RNA, but the DNA is special because it can't leave the nucleus, and its structure is made up of a tightly wound or coiled double helix that's held together by weak hydrogen bonds. And you can notice that this RNA is actually a single strand, but what it is a blueprint copy of this master copy of DNA that is going to leave the nucleus and go to the ribosomes for protein synthesis. So both are needed in order to make the proteins, but only the RNA can actually leave the nucleus. The DNA stays inside. Rosalind Franklin was actually the one that figured out the structure of DNA and figured out that it was, some sort of double helix type shape by using X-ray diffraction in what's called photo 51. And you can see this structure in the middle here. And that was the start of, learning about the structure of DNA. Unfortunately, she passed away before she was able to be credited with it, and Watson and Crick are the scientists that are credited with figuring out the structure of DNA. And we can see that here is what it would look like if we uncoiled this double helix. And the backbone, or these outside strands, remember there's two, they're made up of sugars and phosphates, and then we have a nitrogenous base in the middle. And the strands are held together by the nitrogenous bases by weak hydrogen bonds. So they're going to be able to be broken apart. When we look at a structure of DNA, we're really looking at these long chains of nucleotides put together. Remember, the nucleotides are the building blocks of a nucleic acid, which would include DNA and RNA. And we're going to have specific pairing of these bases. So thymine and adenine, or T and A, are always going to be paired, and guanine and cytosine are always going to be paired. And it has to do with the shape and the bonds that can be formed in between the two. If we look at a comparison of DNA and RNA, we know that DNA is called deoxyribonucleic acid and RNA is ribonucleic acid. And that's because the sugar in RNA is called ribose. It's still a 5-carbon sugar, but that's ribose. And the sugar in DNA is deoxyribose. The structure also differs. DNA is double-stranded and tightly coiled double helix, whereas RNA is a single strand. And DNA's structure is held together by weak hydrogen bonds. We find both in the nucleus, but DNA is only going to be found in the nucleus, and RNA can actually leave the nucleus through those nuclear pores, and that's going to take that blueprint copy from the master copy of DNA and bring it to the ribosomes in order to make proteins. And we find the bases adenine, thymine, guanine, and cytosine in DNA, but we only find the bases adenine, uracil, guanine, and cytosine in RNA. And there's also two types of RNA. We're going to find mRNA and tRNA, which are going to have jobs when we talk about transcription and translation later in protein synthesis. And one thing to remember about the strands of DNA is that they're anti-parallel, so that they complement each other. So it's going to appear that the strands go opposite of each other. That's what anti-parallel is.